Welcome to Base by Base, the papercast that brings genomics to you wherever you are. Thanks for listening, and don't forget to follow and rate us in your podcast app.
Absolutely. Thanks for tuning in.
So, to start off today, I want you to take a second and just think about the last really heavy carbohydrate meal you ate.
Oh, man. Like a giant bowl of pasta.
Yeah, exactly. Or, a thick slice of sourdough, maybe a huge plate of rice. We kind of just take it for granted that our bodies can digest all that starch without a second thought.
It just happens.
But the genetic tools required to process the transplex carbs, they're actually incredibly specific. And, well, the prevailing scientific narrative has long tied the expansion of those exact genetic tools to the dawn of global agriculture.
Yeah, the assumption being that once humans started farming and cultivating these massive cereal crops, our genomes just underwent this huge shift to handle all that dietary starch.
Which brings us to the core mystery of our deep dive today. How could a prehistoric, foraging population possess the complex genetic machinery for a heavy carb diet, like thousands of years before agriculture was even invented?
It's such a crazy puzzle.
It really is. And to solve it, we have to look at the Japanese archipelago. But studying ancient populations in Japan introduces this massive geographic hurdle.
Oh, a total nightmare for paleogenomics.
That's because the soil across mainland Japan is notoriously warm, it's humid, and it's highly acidic. So when you're looking for ancient DNA, that specific environment acts like a biological incinerator.
It breaks down genetic material so incredibly fast.
Right. So for decades, piecing together the genetic history of this region has been this massive exercise in frustration. It's sort of like trying to read a manuscript that's been left out in the rain for a few millennia.
Exactly. The history is buried there, but the Earth itself is actively erasing the data because paleogenomics entirely relies on preservation.
And humid, acidic environments are basically the worst-case scenario.
The absolute worst. Which sets up this incredible locked-room mystery for geneticists. Like, how do you uncover a population's secrets when the environment is actively destroying the evidence?
And that is exactly what makes today's paper so exciting.
Yeah, today we are celebrating the work of an amazing research team who actually managed to bypass that environmental destruction. They generated some truly stunning data.
Who are we talking about here?
We're looking at a landmark paper led by researchers Koji Ishiya, Fuzuki Mizuno, Jungojo Bori, Masaiku Kumagai, and their colleagues.
Awesome.
And it was this massive collaboration across multiple institutions, including Sapiens Life Sciences, Kanazawa University, Toho University, and the National Agriculture and Food Research Organization in Japan.
A really heavy-hitting team.
Truly. We are celebrating their work today because they successfully generated high-coverage ancient genomes that have fundamentally advanced our understanding of prehistoric migrations, population structures, and dietary sifts in East Eurasia.
So to really grasp the magnitude of what they did, we should probably ground ourselves in the timeline of Japanese prehistory.
Yeah. We need to set the stage. We're looking at two defining transformative eras here.
Okay, lay them out for us.
First is the Jomon period. This spans from roughly 16,000 years ago up to about 3,000 years before present.
Wow, so a really long stretch of time.
A massive stretch. And the Jomon are fascinating because they completely defy our typical expectations of hunter-gatherers.
Oh, so?
Well, instead of being highly nomadic, they established this early sedentary lifestyle. They built complex settlements and even created some of the oldest known pottery in the world.
Right, so they were foragers, but they stayed put.
Exactly. And then following the Jomon, you have the Yayoi period. That runs from about 3,000 to 1,700 years before present.
And this is when everything changes, right?
Completely. This era represents a huge cultural, technological, and demographic pivot. It's characterized primarily by the introduction of paddy rice cultivation, which came over from the Eurasian continent.
So the landscape basically transitions from these deeply rooted, settled foragers to a full-blown agricultural society fueled by continental migration.
But it's a shift, yeah.
And obviously that kind of transition involves massive biological restructuring. But because of that DNA-destroying soil we mentioned, previous genetic research was pretty constrained, wasn't it?
Extremely constrained. The field was mostly working with low-coverage genome data or they were isolating just mitochondrial DNA.
Which only gives you the maternal line.
Right, exactly. Mitochondrial DNA is great for tracing broad maternal lineages, but it just doesn't give you the full autosomal picture.
You're missing half the story.
More than half, really. And the low-coverage nuclear genomes that were available, they simply lacked the statistical power required for deep mechanistic insights.
Because when your coverage is low, you can't perform precise diploid genotyping, right?
Exactly. You can't confidently read both sets of chromosomes inherited from the mother and the father, and you certainly cannot reliably measure structural variations like gene copy numbers.
And those gene copy numbers turn out to be the most critical part of this entire study.
I always think about low coverage data like a highly pixelated standard definition photograph.
That's a great way to put it.
Like you can make out the broad shapes, you can see a forest, maybe a mountain, a blurry person standing there, but you cannot count the leaves on the trees or see the texture of the person's clothing.
Right, the fine details are totally lost.
But what this research team accomplished is essentially an upgrade to a 4K ultra-high definition image. They brought the genetic features into such sharp focus that we can now see individual evolutionary adaptations.
But achieving that 4K resolution meant they had to bypass the acidic soil problem entirely.
So how did they do it?
The leap in resolution really started with careful sample selection and a targeted extraction strategy. The team focused on two specific ancient individuals.
Okay, who were they?
The first is an initial Jomon female, designated IY1. She was excavated from the II rock shelter site and dates back to around 8,300 to 8,200 years ago. So she is situated deep within that early hunter-gatherer era.
Exactly. And the second is a middle Yayoi male, designated Dio, from the Doigahama site. He dates back to around 2,300 years ago.
Placing him right in the middle of that agricultural transition.
Right. And to retrieve usable DNA from these two, the researchers targeted the petrous bones.
The petrous bone. This is one of those brilliant biological workarounds.
Yeah, it really is.
For you listening, if you picture the base of the skull right near the inner ear, there is this pyramid-shaped portion of the temporal bone called the petrous part. And it gets its name for the Latin word for rock.
Because it is the densest bone in the mammalian body.
It's incredibly tough.
Yeah, and that sheer density creates this microenvironment inside the bone that is highly resistant to external degradation, bacterial infiltration, all that chemical weathering.
It acts like a biological black box flight recorder.
Oh, I love that analogy.
Like, the external environment might be highly acidic and humid, actively destroying the rest of the skeleton, but that dense casing of the petrous bone preserves the genetic flight data safely inside.
Exactly. And by targeting that protected vault, the researchers were able to deploy high-coverage shotgun sequencing.
Shotgun sequencing, that sounds aggressive.
It's a wild technique. Basically, the entire genome is randomly sheared into millions of tiny fragments. The sequencing machines read these short fragments completely blindly.
And then what? How do they make sense of it?
Then massive computational power is used to map them against a reference genome, finding the overlapping ends to stitch the entire sequence back together.
So it's super computationally heavy.
Extremely. But it gives you a comprehensive look at the entire nuclear genome rather than just looking at targeted markers.
But the really staggering metric here is the depth of coverage they achieved on those fragments, right?
It is mind-blowing. They hit 67-fold coverage for the 8,300-year-old Jomon individual.
6-7-fold.
And 46-fold coverage for the Yayoi individual.
Okay, let's break that down for people. In genomics, coverage indicates the average number of times any given base pair in the genome has been sequenced.
Right, so if you have one-fold coverage, you've seen a piece of code exactly once.
Which means if there's a mutation, you can't mathematically prove if it's a real biological mutation or just a glitch in the sequencing machine.
Exactly.
Yeah.
But at 67-fold coverage, they have read the Jomon genome independently 67 times over.
That is insane for an 8,000-year-old sample.
The statistical confidence in every single genetic variant is essentially absolute. It's the kind of precision you'd expect from sequencing a living patient in a modern clinical setting today.
Which naturally introduces a really heavy dose of skepticism, doesn't it?
Oh, absolutely.
Because if a paleogeneticist is looking at DNA pulled from the dirt after 8,000 years, and the sequence looks as clean and complete as a modern clinical sample, the immediate fear is contamination.
It's the specter that haunts all ancient DNA research.
Like, how do we know we aren't just looking at the DNA of the modern archaeologist who happened to sneeze on the petrous bone in 1980?
It's a very real concern. But to prove authenticity, the researchers didn't just look at the code itself. They looked for chemical damage that is strictly specific to antiquity.
Ah, right.
They measured what are called post-mortem deamination patterns.
The mechanism behind deamination is actually a brilliant piece of chemistry.
It's fascinating.
When an organism dies, its cellular repair mechanisms shut down entirely. So over millennia, the DNA backbone fragments in the exposed ends undergo this predictable thermodynamic reaction.
Right. The water in the environment basically attacks it.
Yeah, the cytosine molecules, the Cs in the DNA code, are attacked by water, causing them to lose an amine group. And when cytosine loses that amine group, it chemically converts into uracil.
And because this hydrolysis happens almost exclusively at the frayed ends of the fragmented DNA strands over, vast stretches of time, it acts as an unforgeable microscopic timestamp.
Because modern DNA does not have cytosine to uracil conversions localized at fragment ends.
Exactly. Modern DNA is intact. So by verifying these exact predictable damage patterns, the team proved the sequences were authentically ancient.
And what was the contamination level?
They mathematically calculated that both individuals had less than 1% modern DNA contamination.
Wow. Okay, so with the data authenticated and sitting at that ultra-high 4K resolution, the team could finally apply advanced analytical tools to reconstruct the demographic history.
And the main technique they used to look back in time is known as a PSMC analysis.
Okay, PSMC, what does that stand for?
It stands for pairwise sequentially Markovian coalescent.
All right, that is quite the mouthful.
It really is.
Yeah.
But it's a powerful statistical method that allows researchers to estimate historical population sizes using only a single high-coverage genome.
That concept can be incredibly counterintuitive at first glance. Like, how does a single person's genome tell you how many thousands of people were in a population tens of thousands of years ago?
It sounds like magic, but the mechanism relies on heterozygosity.
Right.
Every individual carries 2 copies of the genome, one from their mother and one from their father.
Right, we get half from each.
So by traveling along the chromosomes of this one individual and measuring the density of mutations, basically the spots where the maternal and paternal lines differ algorithms can estimate how long ago those 2 specific ancestral lines shared a common ancestor.
Oh, I see. So if the population in the past was very large, it takes a long time going back into history for those 2 lines to finally meet.
Exactly. But if the population was small, they coalesce much more recently.
So the single genome is literally acting as a massive ledger of historical genetic diversity.
Precisely. And when they ran the PSMC on these 2 individuals, they looked specifically at the demographic trajectories after the last glacial maximum, when the ice sheets began to retreat.
And what did that reveal?
The analysis revealed starkly diverging demographic paths. For the Jomon lineage, the estimated population size remained remarkably stable and flat.
Really? No post-Ice Age boom?
None. There was no explosive growth or sudden numerical expansion following the end of the Ice Age. They simply maintained a steady presence in the archipelago.
And the Yayoi?
Conversely, the ancestral population of the Yayoi individual exhibited a gradual, sustained, and significant increase in size over that exact same post-glacial period.
So those diverging growth trajectories perfectly mirror their distinct ancestral origins.
Yes, they do.
Because the high-resolution data confirmed that the Jomon lineage was deeply indigenous, right?
Right, very deep. It represents a basal, early diverging branch within East Asian populations, showing profound long-term stability and virtually no genetic influx from outside populations for millennia.
Just totally isolated. But the Io individual, D.O., carried a completely different ancestral signature.
Right. His genome featured distinct continental ancestry, with specific genetic components linking back to ancient Northeast Asian populations, particularly around the Amur River and West Liao River region.
Which makes sense for the introduction of agriculture.
Exactly. He possessed genetic markers that were entirely absent from the Jomon individual, which firmly confirms the migration of continental farmers into the archipelago.
But, I think the most compelling part of this deep dive isn't just the map of who migrated where, it's the molecular record of how they actually survived.
Oh, this is the best part.
This brings us to a specific gene known as AMY1.
Right, AMY1. This is the gene responsible for encoding salivary amylase.
Amylase is the enzyme in your saliva that initiates the digestion of complex starches. It starts breaking them down into simple sugars. The absolute second food hits your mouth.
Yeah, if you've ever held a piece of plain bread in your mouth for a minute and noticed it starting to taste sweet,
Oh yeah.
that is salivary amylase actively dismantling the starch molecules on your tongue.
And the really interesting thing about the AMY1 gene is that it is subject to massive copy number variation.
Yes, which is unusual. Most genes in the human body exist in just two copies, one maternal, one paternal. But through evolutionary history, the region of the chromosome containing AMY1 has undergone repeated duplication events.
So some people just have more copies of the gene.
Exactly. And generally speaking, the more copies of the AMY1 gene an individual possesses, the higher the concentration of amylase in their saliva.
Which directly translates to a greater capacity to extract energy from a heavy carbohydrate diet.
Precisely. And because AMY1 involves these repeated structural duplications, you absolutely need high coverage data to accurately count them.
Because low coverage just gives you a blurry mess in this region of the genome.
Right, you can't count the copies if the image is blurry.
So when the researchers finally counted the copies in the Yayoi individual, what did they find?
They found he had about 10 copies of the AMY1 gene.
10 copies. Which makes total sense. A high copy number in the Yayoi individual aligns perfectly with our expectations of an agricultural society.
Yeah, the Yayoi culture introduced wet rice farming, meaning their diet was overwhelmingly starch-heavy. High AMY1 copy numbers would have provided a massive metabolic advantage.
But this is where the revelation happens, the one that forces us to rewrite the textbooks. They looked at the Jomon individual.
Yes, the data showed that this 8,300-year-old hunter-gatherer had about 9 copies of the AMY1 gene.
9 copies.
Yeah.
For a hunter-gatherer.
It's incredible. And to place that number in its proper context, we really have to look at ancient European hunter-gatherers from roughly the same period.
How many copies do they have?
Individuals like the famous Librano 1 specimen from the Mesolithic period typically had only around 5 copies of the AMY1 gene.
Okay, this is where the timeline completely contradicts the established narrative.
It turns it upside down.
Because the prevailing theory in evolutionary biology, which was largely driven by data from Western Eurasia, has been that massive expansions in AMY1 copy numbers were a global, synchronized genetic response to the invention of agriculture.
Right. The logic was that before farming, humans ate primarily meat, fish, and low-starch wild foods, so they just didn't need excess amylase.
So the Jomon genome fundamentally disrupts that West Eurasian-centric model. Here, we have a population living thousands of years before the arrival of rice paddies, possessing an AMY1 copy number nearly identical to later agriculturalists.
It's amazing, and it's crucial to clarify the evolutionary mechanism at play here, because we aren't talking about Lamarckian evolution, right?
Oh, absolutely not. The Jomon didn't consciously alter their DNA by just deciding to eat specific foods.
Right. The mechanism is entirely Darwinian.
During meiosis, the cellular division that creates sperm and egg cells chromosomes sometimes misalign, causing unequal crossing over.
Which results in random duplication mutations.
Exactly. So an offspring might accidentally inherit an extra copy of the AMY1 gene.
Those mutations are random, but natural selection is not.
Boom. That's the key.
The Jomon environment heavily selected for individuals who possess those extra copies, because archaeological evidence shows that the Jomon diet was actually incredibly rich in complex wild carbohydrates.
Right, they weren't just eating deer and fish.
No, they were intensely foraging and processing native plant resources, particularly walnuts, chestnuts, acorns, and early domesticated adzuki beans.
So in a harsh pre-agricultural environment, caloric efficiency is literally a matter of life and death.
Absolutely.
If a random genetic duplication gave a Jomon individual an extra AMY1 copy, allowing them to extract just a little more glucose from a handful of acorns during a brutal winter, they had a distinct survival advantage.
And over thousands of years, that localized environmental pressure drove the baseline copy number in the entire population up to 9.
Which demonstrates that genetic adaptation for high-starch diets occurred independently in East Asia, driven entirely by indigenous plant resources long before continental rice agriculture ever arrived.
It was not a uniform global reaction to farming. It was a highly localized, independent adaptation to their native ecology.
So when the Yayoi people eventually migrated to the archipelago, bringing their continental agriculture in their own high AMY1 copy numbers,
they encountered an indigenous population that was already genetically optimized for heavy starch consumption.
wow. And the genetic blending of these two populations forms the basis for the modern demographic landscape in Japan, a concept known as the dual structure model.
Exactly. The high coverage data provides immense statistical support for this model, which argues that present-day mainland Japanese populations are a multi-stage biological mixture of the indigenous Jomon lineage and the continental Yayoi lineage.
But beyond just confirming the mixture, the researchers used the data to calculate precisely when this admixture occurred, right?
They did, using a technique based on linkage disequilibrium.
Linkage disequilibrium. That is another brilliant molecular clock.
It really is.
When two distinct populations first meet and have children, those first-generation offspring inherit entire unbroken chromosomes from each parent.
Right, big long blocks of DNA.
But with every subsequent generation, a process called recombination occurs.
During reproduction, the parental chromosomes cross over and swap segments, effectively chopping those long blocks of ancestral DNA into smaller and smaller pieces.
So by measuring the length of these shared intact segments in a population today, geneticists can mathematically work backward.
Right, to determine exactly how many generations of chopping have occurred since the populations first mixed.
And what do they find?
The researchers measured this decay in linkage disequilibrium and estimated that the primary admixture between the Jomon and Yayoi lineages occurred roughly 65 to 79 generations ago.
Wow. So it really paints a picture of a dynamic, multi-stage integration rather than a sudden total replacement.
Yeah, the continental ancestors likely experienced admixture on the Eurasian mainland before migrating, followed by a profound, sustained period of integration with the Jomon once they were within the archipelago.
It is an incredibly detailed rendering of human history, pulled from just a few fragments of bone.
It is.
But as we synthesize all this, I think it's important to acknowledge the limitations that the authors themselves highlight in the paper.
Absolutely, we have to keep perspective.
Because while the depth of the data, 67x and 46x coverage, is an absolute triumph for paleogenomics, the study is ultimately anchored by just two individuals.
That's the big caveat. The Japanese archipelago spans a vast geographic and climatic range.
Right, from the severe snowy winters of Hokkaido in the north down to the subtropical environments of the southern islands.
So two genomes provide a spectacular baseline for the initial Jomon and middle Yayoi periods, but they simply cannot capture the full spectrum of regional diversity.
Which means future sequencing efforts will need to uncover more high-coverage genomes across different islands and distinct chronological phases.
Right. We need to see if these high AMY1 copy numbers hold true universally across all Jomon settlements, or if there were isolated pockets where the dietary pressures and therefore the genetic adaptations look different.
The resolution of the picture will only improve as more data emerges. The core discovery presented in this paper remains a total paradigm shift for how we view dietary evolution.
It completely changes the conversation.
So, to bring all this together into a central takeaway for you listening, these high-resolution ancient genomes reveal that human genetic history isn't solely defined by massive, sweeping continental migrations or the global invention of agriculture.
No, not at all.
Our DNA is equally shaped by deep, localized environmental stability and the highly specific native plants our ancestors managed to forage. The Jomon people underwent a profound genetic adaptation, fine-tuning their biological machinery to extract energy from the acorns and chestnuts of their native forests thousands of years before the first grain of rice was planted in Japanese soil.
It really forces us to appreciate just how intimately our genomes are tied to the local ecology of our past.
It does. And it leaves us with a really fascinating thread to pull on. If a localized diet of wild nuts and beans can exert such intense evolutionary pressure that it leaves a permanent structural signature in the human genome, what does this mean for our understanding of how our own highly processed modern diets might be actively shaping the genetic legacy we leave for future generations to uncover?
That is a wild thought to leave on.
Definitely something to chew on. This episode was based on an open access article under the CC BY 4.0 license. You can find a direct link to the paper and the license in our episode description. If you enjoyed this, follow or subscribe in your podcast app and leave a 5-star rating. If you'd like to support our work, use the donation link in the description. Now, stay with us for an original track created especially for this episode and inspired by the article you've just heard about. Thanks for listening, and join us next time as we explore more science, base by base.
whispers in the night. After the ice, the pathways split. One steady flame, one rising fear. Names are gone, but the signals stay in every marker of buried day. Hey, call it soul in a double helix. Call it drums in the data we mix. We were many roads, we were one home, and the starch was singing before the grain was grown. Clap your hands and let the past come through. We taste tomorrow in what was true. One line holds fast like an anchored shore. One line surges a crowded door. A continental tide in the new world blood. Threads of continuity under the mud. Count the copies, taste the clue. 9 or 10 and it's ancient too. Before the patties, before the rice,